To examine phytochrome responses in specific plant cell types, GAL4-GFP enhancer trap and UAS-BVR transgenic lines are crossed to produce phytochrome deficient UAS-BVR cross GAL4-GFP progeny. Leaf protoplasts are isolated from plants. Next, the protoplasts are sorted by FACS to isolate GFP negative and GFP positive protoplasts in separate samples. Isolated protoplasts are analysed by confocal laser scanning microscopy to confirm the collection of GFP positive protoplasts. RNA is extracted from protoplasts and analysed spectrally.
Hi, I'm Sankalpi Warnasooriya from the laboratory of Beronda, Montgomery in the Department of Energy Plant Research Laboratory at Michigan State University. Today we will show you a procedure for isolating leaf protoplasts from Arabidopsis thaliana for fluorescence-assisted cell sorting and cell type-specific gene expression analysis. We use this procedure in our laboratory to study tissue and organ-specific phytochrome-dependent responses. So let's get started.
The Arabidopsis thaliana line used in this procedure was generated by a bipartite enhancer trap approach. Parent 1 is from a library of enhancer trap lines with different patterns of GAL4 expression. Parent 1 also expresses GFP under the control of an upstream activation sequence, or UAS in short. The transcription factor GAL4 activates any gene placed under the control of the UAS. In the parent 1 line, GAL4 activates GFP. Parent 1 was crossed with parent 2. In parent 2, the BVR gene is under the control